Okay, so where we left off again was at the section on thiols, and this is a really brief section. And as I told you last lecture, I think that by now you all know that when you have an R and a hydroxyl, that's called an alcohol. So when you take an R group and you put an SH on the end of it like that, that's called a thiol. So it says similar in structure to alcohols, except you just replace the oxygen with a sulfur. And first, we're going to look at how to name thiols, and it says the name is based on the longest alkane chain with the suffix thiol, and the position will be indicated by a number. So very close to the nomenclature of alcohol. So if you understood that, you should not find naming thiols too troublesome. Here are some examples of different thiols. So again, if you had this alcohol, CH3, CH2OH, that would be called ethanol. And so what you do is you take ethane. So if you had CH3, CH3 like that, since you're removing one of the hydrogens, you take ethane and then you add the word thiol on the end. So this becomes ethane thiol. And for example, here, if you had two thiol groups, one on each carbon here like this, that would be 1,2-ethane dithiol. So that would be a dithiol. Here's another example. You can see that the longest carbon chain contains 4 carbons, so that would be butane. You have a thiol at carbon number 1, so that's 1 butane thiol. And then we have a methyl in the third position, so that's 3 methyl, 1 butane thiol. Here's another example of a dithiol. We take the exact same approach. We find the longest carbon chain. This time, the longest carbon chain has 2 thiol groups on it. Now, where are you going to start? Are you going to start here, 1, 2, 3? No, you would actually start from the other end like this, 1, 2, 3, 4, because you want to give the first thiol group the lowest possible number, and that would be the number 1. So this becomes 1,4-pentane because we have 5 carbons in the longest chain, 1,4-pentane dithiol. So that's how to name thiols and dithiols. Let's keep moving on. Something else that's interesting about thiols is they have very unpleasant aromas. It says here that they are even nauseating. For example, if you've ever smelled skunk spray, or you can see that the spray of a skunk, they actually have highlighted a couple of the compounds that come from a skunk. So you can see here the trans-2-butene, and that's a thiol. You can see the functional group there. 3-methyl-1-butane thiol, that's another one here. In fact, that was yeah, so those both come from the skunk, and then I don't know why, I always thought this was kind of a funny picture. She actually compares them to the structures of some odors that come from geraniums. So you have this compound here, which is an alcohol, which again is a geraniol, and then you have 2-phenyl ethanol, which is another compound that gives geraniums their odor, their pleasant odor. Anyhow, so you can see that there's a big difference between a thiol and an alcohol. Huge difference. In fact, working with thiols is really tough. I've done that a few times in my life, and they're so stinky that if you drop anything in the lab, if you spill a drop or anything, you'll usually become very unpopular with your lab mates. Anyhow, let's move on from here. Talk about disulfide formation. This is something that we'll look at even more later on in the class when we do a chapter on amino acids. But I just want to take a quick look at this reaction where you have two. These are 2 amino acids. So the name of the molecule isn't 2-cysteine. It's just there's 2 cysteine molecules. So this is a cysteine molecule right here, and then this is another cysteine molecule here. And these are both amino acids, and they're both identical, right? So these are amino acids. And what it's saying here is it says the thiol disulfide redox pair controls a critical factor in protein structure called a disulfide bridge. And later on, we're going to do a chapter completely about proteins, and we're going to talk about disulfide bridges in a lot of detail. So when you take 2 cysteine molecules, which are again both amino acids, they can undergo oxidation to form this molecule here, cysteine. Okay, so what it's saying here is the thiol. So a thiol, as we discussed earlier, is RSH, and a disulfide is when you have two sulfurs in between two R groups like that. So here's the disulfide. What I want you to know for the next exam is that when you take two thiols and when you make a disulfide bond from two thiols, that's an oxidation, okay? How do you know that that's an oxidation? Why? Because you end up losing, you're losing hydrogen, right? You're losing hydrogen. And as we discussed previously in this class, in organic chemistry, oxidation is characterized by either gaining oxygens, which you're not doing here, or losing hydrogens. Conversely, if you look at the reaction in the opposite direction, we take the disulfide and we reduce it to the two cysteines. Well, what the heck is going on here? That is going to be oxidation. Oxidation, because we said that oxidation is either, sorry, it's going to be reduction. I'm getting confused here. It's going to be reduction because we're gaining hydrogen. So we'll put that in here, gaining hydrogen. There we go. So you should know two thiols to make a disulfide is oxidation. And when you break it apart to make two thiols, that's reduction. Okay, nothing more than that. And you should also know what a disulfide is. I just went over that with you. A disulfide. There we go. And then the whole thing about protein structure, why don't we save that for the chapter on proteins, right? And here's just an example. This is a slide that discusses briefly the structure of insulin. And you probably don't even know what all these little spheres are yet. These all represent different amino acids all linked together. So again, we'll talk about that in the chapter on proteins. But the important thing is that you know that in insulin, there's a couple of disulfide bonds that are shown here. Anyhow, and I won't ask you anything about that, I don't think. Anyhow, so this is just a summary of what we covered in chapter 12. We discussed reactions of alcohols. We talked about the hydration of alkenes, and you have to know Markovnikov addition. Then we talked about the reduction of an aldehyde or a ketone, and we said that when you take an aldehyde and you reduce it, it makes a primary alcohol. And when you take a ketone and you reduce it, you get a secondary alcohol. What else? We talked about reactions of alcohols, and I said that you have to understand what a Zaitsev product is, and you should be able to identify or draw a Zaitsev product. Then we talked about oxidation, which is the opposite of reduction that we looked at here. And then a primary alcohol gets oxidized to an aldehyde, which can also be further oxidized to a carboxylic acid. Carboxylic acid, that can happen. And then it says here, secondary alcohol gives you a ketone, and then a tertiary alcohol doesn't react whatsoever. We also talked about the dehydration synthesis of an ether. That's where you take two alcohols and you combine them with acid, a catalytic amount of acid, and you heat that up. So I'll write down here heat, okay? And then you form an ether like this, plus you drive water from the reaction. What else? And this is just a slide that shows you all the different reactions. And so I'm not going to spend a lot of time on this since I just went over this, but I'll just put this bug in your ear. Make sure that you're, really well aware of Markovnikov addition, okay? And make sure you're really well aware of Zaitsev product formation. You can identify the major product of a dehydration reaction. All right. And then here are some practice problems that you can look at in our textbook. Okay, well, that covers all of chapter 12.